Hi everybody, it's a pleasure to be with you today, even if we would have preferred, like everybody, to meet personally in Rome as it was originally scheduled. So I will update you on our program on micro-dystrophin development as a gene therapy for Duchenne, and also give you some information on how our association is working, what is our strategy. The AFM-Tėlėthon in the French Parent Association, non-for-profit association, founded more than 60 years ago by patients and parents of patients. And the objective of the AFM-Tėlėthon is to find cure for rare diseases in general and including neuromuscular disorders like Duchenne. We are an organization combining volunteers and also employees with the only goal to find treatment for these progressive and devastating diseases. The AFM-Tėlėthon is funded thanks to a special weekend we organize, usually the first weekend of December, which is very important for us because during these 2 days we have a 30 hours live TV show on the main channels of the French television. You can see some picture on the screen. And also we organize several thousand local festive events in most of the cities in France, where volunteers are organizing these events and the French citizens are participating and donating money for our actions. So thanks to these 2 days, we raise more than 90 million euros per year to support all of our actions. We decided early to have our own laboratories. That's why we created, back in 1990, the GNT0004 laboratory, which is dedicated to the gene therapy products for neuromuscular disorders and other rare diseases. And then 7 years later, we created the Institute of Myology, which is a translational research center that has both a research activity on neuromuscular disorders, but also is a clinical trial center involved in many clinical trials. And recently, we created the iStem laboratory dedicated to cell therapies. All in all, this task force is composed of more than 600 scientists working on the development of biotherapies. And we have a budget of more than 60 million euros for research in all these 3 research centers. Our support to research is not only focused on basic science in France, but also outside of France, but also to help the development of the products through a seed fund that we organized to help startup companies to develop their products. And then more recently, we decided to start our own pharmaceutical company called Hypothesy. Hypothesy is a fully operational pharmaceutical company dedicated to gene therapy products. And we also are involved in several industrial partnerships with biotechs and pharma companies to be sure that the projects we supported from the basic science can go to the patients. Okay, and now I will tell you a few words of our gene therapy product for Duchenne, which is the development of a micro-dystrophin. You all know very well that micro-dystrophin is the only way to express a dystrophin in a gene therapy, as the full-length dystrophin is too large for the capacities of the viral vectors that are used as gene therapy. This micro-dystrophin is under the control of a specific promoter to express the micro-dystrophin only in the muscle and the heart. The vector that is being used is called AAV8, and this AAV8 vector is the same viral vector that is used in another clinical trial which is ongoing on another myopathy, which is the myotubular myopathy MTM1. And this clinical trial is ongoing with very good preliminary results and also a very good safety, which is mostly important. We first validated the activity of this vector in several animal models of Duchenne muscular dystrophy. The first model for which we validated the activity was in a mouse model called MDX. And as you can see here on this slide, the action of this microdystrophin viral vector is very good, as it normalizes the muscle force and other activities that have been measured in this model. And below you can see also in the muscles of a dog model of Duchenne that the expression of this microdystrophin is very high in the muscle tested, and also that this expression is lasting for several years. And then in this last model of the Duchenne muscular dystrophy, which is the rat model, we validated, of course, the expression of the dystrophin, but we also observe here something which is crucial, which is the reduction of the fibrosis and the reduced connective tissue amount in the muscles. This is only a small number of examples of the results we obtained using this vector in these models. But to summarize, this vector showed a very good expression of microdystrophin in the muscles. It was very well tolerated. The activity is sustained for a long period of time. We observe no cytotoxic immune response to the transgene, and we also observe a reduction of fibrosis. We are now moving to the preparation of a clinical trial. And for this clinical trial, we already determined the minimum efficacy dose that would be injected in the patients. We also completed long-term toxicology and biodistribution studies of the vector. We are organizing an international multi-center prospective longitudinal baseline study on patients, and the clinical trial itself will be multi-center, and we are filling the clinical trial authorization this year to have the answer before the end of the year. And in parallel, we have already obtained the first batches of the product that will be injected in the patient, and this production is still continuing. Finally, we have announced at the beginning of this year that we have reinforced our collaboration with Sarepta company. This is the continuation of a first collaboration that started 3 years ago for the development of this product. And with this partnership, GNT0004 will be responsible for the commercialization of this clinical product in Europe, excluding United Kingdom, and Sarepta will be responsible for the rest of the world. And the company that will manufacture the product is Hyposkesi. It was selected to produce the clinical batches used in the clinical trial, but also for future production of the product. I thank you for your attention, and I will be happy to answer any question that will be forwarded to me. So until then, take care for you and your families. Thank you. Bye-bye.